Thank you very much for joining the Office of Science and Technology Austria for this webinar. My name is Julia Hurley, and I will be hosting this event. Today, we're speaking with 3 scientists from the Albert Einstein College of Medicine about their research on yellow fever vaccines. So we have with us today Dr. Haslwanter, Dr. Chandran, and Dr. Lasso, who will introduce themselves in a minute. So today's event is a unique initiative that was developed because of ASTA's mission to highlight the work of Austrian scientists working abroad and to promote science, diplomacy, and collaboration. Our conversation today will focus both on the team's research and how this international team has worked together. So first, the scientists will give an overview of their work, after which we will have a moderated panel discussion. We ask that the audience members remain muted during this part of the webinar and submit questions either via the chat or once we open the conversation to the audience at the end. So also, as I mentioned, this discussion will be recorded and posted on our YouTube channel and shared on our website. So without further ado, I would like to invite our panelists to introduce themselves.
Okay, good morning or good evening, everybody. I'm Denise Haslwanter. I'm a research fellow in the lab of Dr. Kartik Chandran at the Albert Einstein College of Medicine. And my research is focused mainly on emerging viruses, such as yellow fever virus. And in particular, I'm working on antibodies and vaccines.
Go ahead, Gorka. You're muted.
Yes. So, hi, my name is Gorka Lasso. I'm a research assistant professor at the Chandran lab, and I'm a computational biologist focusing mostly on viral host and viral protein-protein interactions.
I'm Kartik Chandran. I'm a professor of microbiology and immunology at Einstein in New York City. We have a group of about 15 students, research fellows, research faculty, that are working on emerging viruses, how they infect cells, and how to develop ways to block their infection, and also develop vaccines, and some of that we'll talk about today.
All right, thank you very much. So next, I believe Denise has prepared a presentation to introduce us to the work.
Okay, can you see my slides?
Yes.
Okay, I prepared a short presentation about our recent findings. So we were investigating the human antibody response to the yellow fever virus vaccination, and that it displays a reduced potency against a novel circulating Brazilian strain. So yellow fever is transmitted by mosquitoes, and in particular by the yellow fever mosquito known as Aedes aegypti. And if you get bitten by a mosquito which has this virus, you might experience no symptoms, so you undergo an asymptomatic phase, or you might experience symptoms such as sudden fever, yellowness of the eyes, dark urine, which is what also gives name to the disease, as well as body pain, headache, and also bleeding from the nose, ear, and mouth. There are approximately 200,000 infections a year, and 90% of them actually happening in Africa, and only 10% are in South America. Deaths are about 30,000, and the case fatality rate ranges about 5 to 15%. The yellow fever disease can be easily prevented by a vaccination, and the vaccination is safe and also free in many countries in Africa and South America. And the vaccine exists since 1937. It was invented by Max Theiler, and it is a live attenuated virus. And the virus is derived from an African strain, and usually they are called, for example, yellow fever virus 17D, or the substrain 17D204 and 17DD. And here's an example of the typical yellow fever vaccine from Stamaril from Sanofi Pasteur. So the yellow fever vaccine is also thought to be the most successful vaccine ever produced, and it's predicted to give a lifelong protection. However, there are only 35 million doses produced each year, and we would need about 64 million doses to actually cover the need. And therefore, since 2016, and because of continuing outbreaks in Africa and Brazil, fractional dosing was becoming a thing. That means that only like a fifth of the actual vaccine was given to people during outbreaks. So we were in particular interested in South America and yellow fever there, since it is not very well known. Only 10% of the cases are happening there. And what actually caught our interest was this outbreak in Brazil from 2016 to 2019, which was one of the largest one in the past 80 years. So yellow fever in South America is rather new. It started in the Caribbean and then it moved through Venezuela, Colombia, into the Amazonas, where it circulated for several years. But now, most likely because of deforestation and climate change, the yellow fever virus actually moved on down to the south and already by now reached Argentina, but also here, the big capitals of Brazil, such as Rio de Janeiro or Sao Paulo, where these outbreaks are happening. And here you can see the years and or the epidemiological period, the number of yellow fever human cases in Brazil, and the lethality rate, so number of cases are in gray. And there have always been some peaks of outbreaks, but the recent one from 2016, 2019 was clearly the highest with the most lethal cases. So how does these yellow fever virus particles look like? And they look different than what everybody knows now, like the coronavirus, which has the spike. The yellow fever virus particles is completely covered with protein, which you see here. And this protein, which is covering the virus particle, is called envelope protein, or simply E, which forms a so-called homodimer. And this E protein is essential for the virus to attach to a host cell, but also to fuse with the host cell. And thereby, antibodies that are neutralizing are usually targeting this E protein, and thereby also correlation of these neutralizing antibodies means also a protection of the vaccinee. So what we did now to analyze was we wanted to know if the yellow fever strains in Africa and the yellow fever strains in South America, and in particular the one in Brazil, have differences in neutralization. And to study this, we got human vaccinees blood sample from American and from Brazilian donors. So we had 16 from the US and 24 from Brazil, which both received one shot of the yellow fever vaccine. And then we analyzed this for the neutralizing activity, as you see here. And the higher you see the curve is, the better the patients are able to neutralize the yellow fever virus. And here you can see neutralization of these vaccinees against the African strain is very high. However, the neutralization activity against the South American strains by these human vaccinees is up to 90% reduced, so that it's very low. So now we were wondering, why is this the case? And what's the differences between these Brazilian and African yellow fever particles? And we found that on the virus, on this E protein, there are two sites of changes. And here is site 1 in cyan. And site 1 is a novel glycosylation site, which looks approximately what you see here on the E protein in green. This is how a glycosylation site would look like. It sits on the E protein and covers up parts of the particle. The other site, site 2, it's a previously described important epitope where many antibodies are targeting to, and it's very potent in neutralization. However, this site is now missing in the South American yellow fever strains, and thereby again probably reducing the neutralization of the vaccine. So now we were wondering if this was only the case now for this one Brazilian yellow fever strain, or is it for all of South America? So thereby we checked this polymorphism and we compared African and South American strains for this sequence. So here you can see the old world, and they all do have this epitope, which is important for neutralization, but all of them do not have this glycosylation site. And here you can see the smaller ones, the South America 1 and 2 genotypes. So the South American 1 is not having this epitope, but it now has this glycan, and that's for all of them. And that's also where the Brazilian strain is part of. And then there is also South American 2 genotype, which has a little bit of both. It has this glycosylation site, but it also still carries this epitope. So what this would mean in total is that these changes are likely enabling these very important antibodies, which are induced by the vaccine, are now missing or cannot neutralize anymore the South American yellow fever strains to a certain extent. And this work was done with many collaborators at, here at the Albert Einstein College of Medicine, with the lab of Simone Sidoli, with pharma companies, Adimab and MapBio, with the George Washington University, and with the University of Sao Paulo. And in particular, we worked very closely together on this project with Fiocruz in Brazil, with the Bruna Bonallo. She was helping us working on the South American Brazilian virus strain. And that's in short what we did for this project.
Thank you so much for this introduction. It's quite an overview of the work. Yeah, so the next portion of this webinar will be sort of a moderated panel discussion. I will be asking our 3 scientist guests questions, and they will take turns answering, or a couple of them can answer the same question. So my first question to you would be kind of, so what is different about this recent work that you did from the work of other scientists on this topic?
So what is new, I think, is usually our previous studies have been done looking at African strains. So nobody really ever thought much about the South American strains, as they're not considered to be the same, but actually what turned out for us in the end is that they are indeed behaving differently. I think a little bit much how we see now SARS-CoV-2 coronavirus, that actually the more the virus evolves, the more different it gets. And we also need to keep that in mind for the future.
Yeah. Does anyone else have anything to add on that?
Yeah, I'll add that. So there were suggestions that the genetic variants in this new strain might be related to, might be behind the larger outbreak. But like Denise's question on whether it could affect neutralization was a novel question. And yeah, so I think that was one. The hypothesis was novel, and how we addressed it as well by combining both the wet lab and also the computational work to identify the particular residues.
Okay, thank you. Okay, well, I guess then my next question would be, all right, well, with this in mind, what do you see as the direction for future research on this topic? And what could future research?
I can start off with this, and then Denise and Gorka can jump in. I mean, certainly one of the important things that comes straight out of this work is to really sort of think about the kind of antibodies that people normally generate when they're vaccinated with the yellow fever vaccine, and then think about, because we actually don't know what a lot of those antibodies look like, and since antibodies are very important for protection, we want to understand exactly how they're binding to the virus and how that's causing these differences in the reactivity and the neutralization of these African strains versus the South American strain. So I think learning about the antibodies will teach us a lot about the mechanism of protection, but also maybe help us design better vaccines in the future to try and elicit antibodies that can work across all of the yellow fever strains. So that's more on the research and longer term sort of side of things, but there are also some more immediate public health, I think implications for this work that maybe Denise or Gorka, Denise could talk about that. So.
Yeah, I do believe that we kind of need to, even though we think we have vaccines for many viral diseases, we should keep in mind that we still need to evaluate from time to time if they're actually working, how virus is evolving, and things are changing. So it's definitely a public health.
So you would, Denise, you would look into vaccines and try to update something on this?
Yeah, for the yellow fever strain, I think it's worthwhile to think about it, especially since we're not reaching the needed vaccine doses by year. I mean, the virus and the vaccine is basically still produced. The vaccine is still the same way produced as it was almost in 37. So there is definitely a need for changing this, in my opinion, of course.
One more thing that sort of came out of this work that's interesting is just that there are these interesting differences between the African and the South American strains that that actually may be very old. They may go back to the very beginning of how yellow fever got to the New World. And although it's challenging to dig that deep into viral evolution because these viruses evolve very quickly, I think there are some questions to ask about the changes we saw in the South American strains and whether they represent adaptations to a completely new sort of life cycle, different mosquitoes, different primate hosts in the Americas versus what was seen before in Europe and I mean in Africa and in Eurasia. So I think there's some really fundamental questions about how this virus has evolved over the centuries that and what that means in terms of infection and disease and our ability to combat infections that I think are implications that really should be explored in greater detail in the future.
Yeah,
it's like,
from I was going to say, to me, from the molecular standpoint, so we were surprised because we found on site 2, we found this big glycan that it all suggests that helps in blocking antibody binding. But there is a second residue under the glycan that mutates. And when we deconvolute these two genetic variants within the same loop, the glycan plus that single residue, they both seem to affect significantly neutralization independently. So that's super interesting, I think, to try to look into what is it that it's also a hinge that changes conformation, supposed to change, affect conformation of the envelope protein. So I think that's something very interesting to understand how these amino acid changes promote the reduced neutralization.
Great, thank you. I think you touched on this a little bit in your answer because you mentioned public health, Denise. But where do you see any kind of implications for public health policy? Do you see the need for change? Do you think that there will be changes, or how does your research kind of touch on this area? Or can you see a connection to changes in public health policy?
So right now we cannot say from this data that the vaccine is not working in South America or that it works less. There is still neutralization, but the question is, of course, maybe because these neutralization titers are low, maybe you would need to have another booster shot earlier, and maybe it's worthwhile to actually have a better vaccine eventually for this. But one of the most important things would be to test next, does it actually impact the protection, or is it just something we see in vitro, let's say?
It also brings up this issue of what are you actually measuring? So what's done routinely is to measure neutralizing activity. These reference labs, public health labs, use samples from people to look at the amount of antibodies that are neutralizing against yellow fever. But then the question is, what strain of yellow fever do you use for your experiments in the lab to measure this? And traditionally what's been done is to use the vaccine strain. It's the most convenient to use. However, our work really suggests that if you did this in South America, you would be overestimating how much neutralizing antibody there is in the blood of people that have been exposed or been vaccinated than what there actually is, because you're not using the right, what you might call indicator strain of the virus to actually do your laboratory measurement. So your diagnostic test is only as good as the reagent or the way that you're doing the test. And in this case, we would argue that the test should be changed. And there are always complications changing these types of assays, but so there would have to be a lot of work done to really, it's not a trivial thing to change how you do this kind of testing for a whole country. So, but we would suggest that it should be investigated to see if the reference lab should switch from using the vaccine strain as the indicator strain for these measurements, and then switch to using something that looks much more like what people are actually naturally becoming infected with in South America. So right now we think there's a mismatch in how this is done in South America, and that should be addressed at the public health level. So that doesn't speak to how well the vaccines work, but it does speak to how meaningful measurements. Just to give you an analogy that's very, very relevant right now, what's going on with the COVID-19 pandemic. So right now we're at the end of March 20, is it April already? It's April, right? We're in April 2022, time goes by quickly. And in order to look at how much antibodies people are making to the vaccine, for example, that they're getting a booster shot or whatever, which virus are you want to measure the amount of antibodies in the blood of somebody. Which virus are you going to measure it against? Are you going to measure it against the Wuhan strain, which, we haven't seen in 2 years, right? Or are you going to look at Omicron? Are you going to look at BA.1 or BA.2? Are you going to look at a mixture of strains? But it's one thing to do this in a research lab like we do. It's another thing to do this, in a public health lab like hot cakes. So you have to do like hundreds of thousands or tens of thousands of these assays. But if you did Wuhan, you would get one answer. If you did Omicron, you would get a different answer. This is very similar to that. So that in South America, they really should be thinking about using a more relevant strain to do the measurement and not something that isn't circulating in the population, right? We should be looking at what's circulating right now.
Yeah, that's definitely a challenge on a public health policy level to get that level of testing on a broad scale, for sure. No, thank you for that answer. And now, since you've already kind of touched on COVID-19, my next question was going to be, as with everything else, the pandemic has affected kind of every aspect of our life. So my question for you would be, how has COVID-19 affected this research?
Gorka, you want to start? Me?
Whoever, yeah. Gorka, whoever.
Well, I mean, I will start. I think it's the same for all of us in the lab. I mean, it has influenced significantly our research. We're not a lab that works on COVID or coronaviruses, but given the emergency of the situation, we were all trying to help, especially like in our lab, some people work intensively generating clinical assays. And I can tell that most people switch research projects trying to help in. And that, especially even for PhD students, has been complicated because you have your PhD, you need to continue that line of research, but this happened and you feel like this is more important. So it has for most of us in the lab. But now, as things are getting better, we're slowly switching back to our previous projects. But it has remained, and it's still, for some people in our lab, it is becoming a primary line of research.
I think it was very challenging because, although in some ways, we were more lucky than many other labs or people in other lines of work who had to stop what they were doing, or, the kind of work we do, you can't really do it remotely. Well, I could do it remotely, I suppose, because I spend a lot of my time writing, but folks in the lab that are working at the bench can't really. And the problem was during the beginning, the surge in 2020 here in New York in the summer, the spring and summer, we couldn't have the whole lab here because, we had to only have a subset of the people that were here as part of sort of an emergency response to COVID. And we couldn't pack the whole lab into the space. So a number of folks in the lab, about half the lab, had to stay home and work from home, while the other half of the lab came in and only could work on COVID. So it was a very challenging time, I think, for both groups of people, because people that work on COVID, it was kind of it felt like a crisis, and they were working really long hours and trying to help out with clinical projects, like Gorka was saying. But it was, I think, also extremely challenging for people that were stuck at home, couldn't advance their work, and were kind of isolated as well. So it was a very difficult period of time. Fortunately, we were able to get people in relatively quickly. I mean, it didn't feel like that at the time, but I think by the summer, we had people back in lab, and slowly people resumed their previous work. But then, as these variants have come, and we've had different rules on distancing and masking, and this is the one thing in the world that we can safely say that is relatable to like 99% of humanity right now. So I don't have to say, maybe you don't get it because everybody gets it. So we've had the same sort of things, except that we're virologists. So we felt a sense of responsibility and an opportunity to contribute to what was going on as part of the response. But, it also did mean that we had to put what we were doing, which is not COVID, not coronaviruses, on the back burner for a long time. And so, as Denise and Gorka say, it's only now that we're really starting to get fully back to where we were for many projects, including this one. And this is something that the whole scientific community, I think, has experienced, and of course, people in all walks of life.
Did your brief excursion into COVID-19 at all color your perspective when you went back into this research?
Definitely. Yeah. Sorry, go ahead, Denise.
Yeah, no, for the yellow fever research, definitely, because the topics are kind of similar, but it just happened to us by coincidence that they are similar. So we started this yellow fever project way before COVID happened, and then we were actually surprised. And but also, I mean, now this work is also getting more attention because people are aware of the importance of virology now, which is really important for us.
Well, even before COVID, we were a lab that was really, I started this lab 15 years ago working on Ebola. So, we've always worked on emerging viruses. So this is really our thing. And COVID was really the perfect example of, why it was important to work on emerging viruses. But when it actually did happen, the whole scientific community kind of mobilized, in a way that's never really been seen to work on this. And because of that, I think we've learned more about viruses, than, in these last 2 or 3 years than maybe in the last 20 years, just simply because of the concentrated amount of effort that was involved, especially for things like immunology, because, now there were all these millions of people getting infected. And so there were these opportunities to carry out these incredibly rich, studies on the natural immune response to infection, with a focus and an intensity and with numbers of patients and donors. And many people were willing to donate samples for various reasons, to help with the research and to hopefully accelerate developing vaccines and therapies. So our understanding of viral immunology has really dramatically accelerated because of the pandemic. And I think we'll be much better prepared the next time. The other thing that's, of course, happened is the acceleration of the technologies for cutting edge vaccines, like the mRNA vaccines, which have really been a game changer, for the whole vaccine space. And I think we'll see that going forward even more. So yeah, I think when we look back on this, we'll see it as a very challenging, tragic, but also, an incredibly transformative period in, not just human history, of course, but also just in terms of our scientific understanding of viruses and their interactions with hosts. I mean, for us, we had a mini version of this in 2014 with the Ebola outbreak in West Africa, where in a year we learned more about Ebola than we had ever, in the last 30 years before that. And this is, of course, a much bigger version of that. And then one sort of piece is that we just realized how complex the interactions of viruses are with our immune system, and that's a really big part of what we've learned from this pandemic. And that informs this work as well, and lots of other projects.
Yeah. But I also found it very, very exciting to see, like, during these times, that collaborations and all of these things working together changed drastically. Like, everybody was really working, all the scientists towards one goal. Everybody was willing to share samples. Like, and I think that helped a lot to advance the field so much. Because usually, we are working like on one project, one person, more kind of style, and then it changed so much into virologists working together now. Sorry, Gorka.
No, sorry, I interrupted. But I was gonna say on what you are saying, Denise, I think this is moving forward because now as Kartik, we all had to collaborate. We've been working with labs within Einstein and in labs outside Einstein that we never worked with. And this has established collaborations on COVID, but also these collaborations will be carried forward on different projects. So on the positive note on how SARS-CoV has impacted our research, I think we have established new collaborations with people in the clinic and also other basic research labs that we can use these collaborations to further work on our initial biological questions.
Thank you so much for sharing your perspective on that, all three of you. That was really interesting. Well, okay. I mean, so my next question would be, all right, well, we've talked about how COVID has been a major challenge, but in some ways also kind of has helped accelerate your work. But I was curious, kind of what are the other, aside from COVID, what are other major challenges you see in your research right now?
Who's addressing that?
Kartik?
I mean, the challenge is always that science is hard. I mean, nature shows you what she wants to show you, and it's easy to get frustrated, and as a PI, I see this, I get frustrated sometimes, but also, you can see that sometimes it's challenging for people in the lab because you're trying to, not just trying to advance your work but there's a career aspect to it. There's a rat race, right? There's a scientific rat race. And we saw that during COVID as well. And, people don't want to feel like they're not making progress, getting left behind. And, they're also trying to balance this work with the challenges of their lives. And so, I would say, one of the challenging things is to kind of learn about, trying to make, advance our work and learn more about nature. But, balancing that is just, I think for all of us, I mean, we have this love for nature and we really want to understand how nature works and how we all got here and what's going to happen in the future. So it's also an incredible gift and a blessing. But I think the real challenges of this work are in the day-to-day and how you balance all of those imperatives and create have an environment where people enjoy working and feel like they are, doing something important for themselves, for the larger enterprise, for the public, because after all, we do run on public money here. So, and to stay balanced and keep your perspective and not lose your head, in the day-to-day with the challenges of the work. I would say, to me, that's the perennial challenge, but also the perennial opportunity that we have. And we're very fortunate and blessed to be able to do this kind of work.
Yeah, I sometimes felt like you want to do something really meaningful, important for the public, but then you start projects and you're not really sure, is this worth it? Where is it going? Is this actually going to change something, but you're still trying and you're pushing for it? So it's challenging. And also with yellow fever, right now we think it might be important, but if it really is that important, we will figure out later, eventually. So that always feels very challenging as a scientist and frustrating too sometimes, as Kartik said.
To me,
Sorry, go ahead, please.
yeah, no, I was just gonna say, on a different note, but also related to me, a huge challenge that I think we all need to be aware that this can happen again, and we need to put the measures. We need to think a lot about surveillance, and there are things like climate change and globalization that it might push, it might promote this happening again, let's say. So we need to take measures, also quick measures in how to minimize outbreaks when it happen, and try to first anticipate the outbreaks, and secondly try to mitigate the expansion of these outbreaks as much as we can. And it is expected that something like this might happen again, so we just need to be aware of this and take good note of what's happened.
Yeah.
Yeah.
Especially it's just years ago. I mean, it's not so different from yellow fever. It's a mosquito transmitted. It's globalization, as Gorka mentioned. So it could also happen with yellow fever coming to the US. Like the
you prepare for outbreaks between outbreaks. So I mean, it's you can't put things in place while it's happening. Of course, you have to be able to react and be flexible, but like Gorka said, you kind of have to anticipate structures in place, and that goes beyond individual scientists and groups and labs, or even the larger scientific enterprise, because you need buy-in from the governments, the political system, international organizations. You need to build structures to support. I mean, it's appropriate. This conversation is happening under the auspices of the government of Austria, so you know it's appropriate for governments to take steps as well. It doesn't matter how good the science is if we don't have the way to deliver those advances to people at the street level. So you can have great drugs and vaccines, but if you can't deliver them in time to remote places or to penetrate into the population, then it, in some ways, the science from the public health standpoint doesn't really matter, because you know it has promise, but it, you know, in order to deliver on this promise, it needs just this much larger sort of everybody pitching in at various different levels to build structures that are needed. And we saw this a lot during COVID with vaccines and therapeutics. Bodies, but
Oof, sorry about that. My internet connection doesn't seem to be collaborating today. But thank you so much for your insight on that. Well, so one thing I wanted to address was, all right, well, I think the general public has taken a major leap in understanding of kind of the basics of virology and why it's so important. But, you know, as we've seen in the pandemic, there are still major misconceptions. And I wonder if there are any kind of misconceptions about the work that you do in particular, sort of aside from the COVID misunderstandings that we're all familiar with. Are there any misconceptions that you're fighting against right now?
I feel one of the misconceptions, as mentioned before, is that just because neutralization titers are low, that there might be no vaccine protection. So that is not a case. We still have to investigate that, and still if you have low amount of neutralization, that might be still sufficient for protection. And also besides antibodies, there are also other factors in your immune system which work for protection, such as T cells. So this still needs further investigation. And I know it's also a topic that has been discussed with COVID, because people say titers are low, but we cannot really tell if there is no more protection. And even a little bit protection is still better than no protection and not getting vaccinated.
So basically not losing doubt in the vaccines before we understand sort of why things work and why some things don't. Okay. Yeah.
It's the same issue with COVID, right? I mean, it's not just that people don't have misconceptions, it's that those misconceptions are being actively stoked and fueled to divide people by individuals who have their own agendas, whether they're political or economic, disinformation, on social media, among and other places where people get their information and news these days is rife, right? I mean, it's not just that people don't understand or they have misconceptions; they're being actively fed lies, and false information by people who have their own agenda. So, governments, and public entities have to deal with that. Individual scientists are not going to be able to really fix that. I mean, we can be part of the solution, but this is a larger structural problem about how the public gets its information. And, this manifests in every possible way, in our culture, not just with viruses and vaccines, but lots of other things too.
But I also believe it's because we scientists are trained not to say it is like this, we say it might be, while other people in other areas, like especially people who not believe much in vaccines, they would rather go for really strong words. And scientists, also if we talk about global change, we always say maybe in 10 years we have 2 degrees more Celsius in the world. It's just a maybe. So people sometimes, if you don't take that so serious, unless I would tell them in 10 years that's going to happen, and that's it. So the communication.
Well, the problem is if it doesn't happen, then
it's
then you've lost whatever little legitimacy you have, right? So it's challenging. I don't think it can be solved by better SciComm. I think this is a much bigger issue that needs to be addressed, at the level of, sort of fora where disinformation is being pumped into the public. A few scientists, holding a sprinkler aren't going to put fire. So.
Yeah, it is definitely an issue that we continue to face at every level today. So thank you for that. Kind of the last area that I wanted to touch on before we wrap things up and open things up to anyone else in the audience who might have questions is, well, as Denise showed in her presentation at the beginning of this conversation, this is aside from the 3 people who were in this virtual room working on this, it was a pretty big collaboration between international researchers, and I was hoping that you would be able to tell me a little bit about how this came together and kind of your experiences and the challenges and the benefits also of working with such an international and diverse team.
So I can start answering that question. So we started off this project as a collaboration with a pharma company, RINA, which they are making monoclonal antibodies as therapeutics, and we help them screening. And I tried to find something novel for our research, as this is more like a clinical work. And then after this finding, we started to draw in more people. We knew we need to get experts from South America, so Nuno Bonaldo's lab, who then connected us to Esper Callas, who is the one who has patient samples in South America. And it's like, then also Gorka, for example, in our lab, he came in later as he's a computational biologist who gave us a very different idea of our wet lab work usually. So it's like, as you go by project, you realize things that are missing for your work to make it more stronger, to make it more interesting, and you start to reach out to people and to try to get help and to figure out more things. So that's usually how it evolves. And I feel the most challenging is that you kind of all stay focused on one goal, and that you kind of continue a good communication, that you keep everybody in the loop. It's sometimes very challenging if there's so many people with so many different interests.
Well, Denise, you made sure we were focusing on the question too.
Yeah, I made constant meetings, poked people. So it's like it's besides working in the lab, a lot of management to keep the people together.
I have a question for Kartik, actually, because in our lab, I've been in few labs, and this is the most collaborative lab I have encountered. So I want to ask Kartik, is like, what does he look in a collaborator? Yeah.
It's a good question. But I would say first and foremost, I'm looking for somebody that trustworthy at a gut level, that this is some, and trustworthy actually is maybe not the right word. Somebody that I would want to enjoy, that I would enjoy working with, like somebody that I would have fun with, somebody where we could talk about science and like kind of, ideally we have complementary skills and interests, sort of complementary skill sets, but we're both passionate about sort of the same thing in the larger sense. And this is somebody that I could really see myself working with. I could see, you know, having a beer with them, and somebody that I think cares about the right things, which, you know, I don't want to work with somebody that's, I don't think that they, you know, I don't know, like, for me, it's at a very gut level, like, is this somebody that I respect as a scientist, but also somebody I respect as a human being to work with? Because I don't want to work with people who are, you know, like, for example, who don't treat people well, or, you know, I mean, every collaboration is a bit of an adventure, right? Like, and every collaboration involves risk. But I think for me, the biggest thing is to be open to the possibility that whatever we do, other people, and collaborating, I think, also involves giving up a certain amount of control. So there has to be a willingness to accept that, to say, you know, I'm going to give away some control on this project. You know, it makes everything potentially more complicated, doing the work, writing about the work, getting money for the work. But I always go into these interactions with the belief that, you know, giving up control is completely worthwhile, because what's going to come out of it is going to be synergistic for everybody. That we're going to get better papers, more money, better science, and the people in the groups are going to benefit from those interactions. And I think an important part of this as well is, when you guess wrong with this, if you enter into a collaboration that doesn't have those hallmarks, then no matter how good the science is and no matter how fancy the papers are, you have to ask yourself, is this worth doing, and should you end it? So I think for me, starting collaborations comes, a lot of it's from gut, but so does ending them, which I think is the other important part of it. But I'm always open to being surprised.